Hello everyone, I am S.V Gaikwad. I welcome you all in the B.Sc paper, that is Genetics and Evolution. So friends, today we are going to start Unit 5th, that is Linkage and Crossing Over. So let's start. So in today's session, we are going to complete the introduction of linkage, its definition, coupling and repulsion hypothesis, then what are different types of linkage, what are the linkage groups, and finally what is the significance of linkage. So let's start. So friends, we know that genes determine the character of an individuals. Okay, and these genes are present on the chromosomes. So on chromosome, these genes, they may be situated closely related or they may be distantly apart. So when these genes are situated on different chromosomes, they assort independently according to Mendel's laws of independent assortment. So there were two scientists, Batson and Punnett, in 1905 observed that the two pairs of alleles they do not assort independently in case of sweet pea plant. Similar results were obtained to Morgan. In 1910, he found that the same phenomenon in Drosophila. So, if the genes are situated on same chromosomes and if they were fairly close to each other, so there is a tendency of these genes to be inherited together. So, these type of coexistence of two or more genes to inherit together is called as the linkage phenomenon. Coming to the definition of linkage, so it is a tendency of genes to remain together in their original combination during the transmission of character. Okay, so this linkage phenomenon was discovered by scientist T.H. Morgan, and this linkage is also reported in Drosophila, then sweet pea plant, then maize plant, and in man also. So there are another phenomenon, that is linked genes. So these are the chromosomes. Okay, so there are different types of chromosomes. On that chromosomes, genes are present. So here the first case of chromosomes, this is the gene first and this is the gene second. So these two genes, they are closely located on the chromosome. So when these genes are closely located, okay, so there is a tendency of these genes to inherit together in the next generation also. So when the genes located on same chromosomes and they inherit together, such type of genes are called as linked genes. So here in the first case, gene first and gene second, they are the linked gene because they show the phenomenon of linkage. The second case is there are two chromosomes. So on these, gene first is present and in that chromosome gene second is present. So these genes, they are present on different chromosomes and there is no possibility of the linkage. Okay, so in that case these are not the linked genes, so they are unlinked genes. Okay, and whenever there are unlinked genes, there is a possibility of crossing over. Okay, the third case is the gene first and gene second both are present on same chromosome, but they are distantly apart from each other. In that case also, there is no linkage possible. So these two genes are also unlinked genes. So linked genes are nothing but when the genes are located on same chromosomes or different chromosomes, but they tend to inherit together at that time. They are called as linked genes. Okay. Coming to the characters of linkage. So a linkage involves two or more genes and they should located in the same chromosome in a linear fashion. The important thing is in a linear fashion. So linkage may involve either dominant genes or recessive genes or some dominant genes, some recessive genes. Linkage generally involves those genes which are closely related. Okay, so presence of linkage leads to the higher frequency of parental types rather than recombinants in the test cross. Okay, more the linkage, more the parental combinations obtained in the test cross. Whereas the strength of linkage depends upon the distance between linked genes. Okay, lesser the distance between linked genes, higher the strength of the linkage. In this way, lesser the distance between linked genes, higher the strength of linkage. So this is a relation between distance and the strength of linkage. So these are certain characters of linkage. The next is coupling and repulsion hypothesis. So this is one of the important hypotheses behind linkage. So Bateson and Punnett formulated this hypothesis to explain unexpected F2 results of dihybrid cross. Okay, so in that there are two phases. First is coupling phase and second is repulsion phase. Okay, first we will see what is the coupling phase. Coupling phase means when the dominant alleles present on same chromosomes and recessive alleles also present on same chromosome, they show coupling phase. For example, in this example, the dominant alleles, that is capital T, capital R, okay, and the recessive alleles, that is small t, small r. So in this way, the two dominant alleles and the two recessive alleles, they show coupling phase. Okay. The next is dominant alleles of some genes. They are linked to recessive alleles of other genes and they show repulsion phase. Okay, they show repulsion phase. Let's elaborate this coupling and repulsion hypothesis by explaining the test cross. Okay, so this coupling phase present in Lathyrus odoratus. Okay, Lathyrus odoratus. So Bateson and Punnett did a test cross in Lathyrus odoratus. So Bateson and Punnett, they made a cross between blue flowers and long pollen grains with red flowers and round pollen grains. So these blue flowers and long pollen grains denoted as capital B, capital B, capital L, and the red flowers, round pollen grains denoted as small b, small l, small l. So these are the gametes. So when they crossed these two varieties in F1 generation, they got hybrid blue flowers with long pollen grains. This is as usual according to Mendel's laws. Okay, dihybrid cross. After that, they made a dihybrid test cross. So later on, when these F1 hybrid individuals, that is hybrid blue long, when test crossed with the double recessive parent, that is red flower and round pollen grains. Okay, so when they crossed these, they got the F2 generation with the 4 types of offspring as usual: blue long, blue round, red long, red round. So in F2 generation, the expected ratio of dihybrid test cross is 1:1:1:1:1. We know that. Okay, dihybrid test cross ratio is 1:1:1:1. But B.Sc and Punnett, they found instead of 1:1:1:1 ratio, they found 7:1:1:7 ratio, and this is unexpected. Okay, so what is the reason behind that? They didn't know. Okay, so in this case, the 7:1:1:7 ratio clearly indicated that there was a tendency in the dominant alleles, that is capital B and capital L, to pass together into the next generation. For example, in this F2 test cross ratio, the parental combinations, that is blue long and red round, these are the parental combinations, and frequency of parental combination is more. Okay, frequency of parental combination is more, so they show 7 ratio. Okay, instead of normal 1 ratio. So here the coupling phase means the tendency of dominant alleles to pass together in the next generation. So these tendency of dominant alleles or recessive alleles to inherit together in the next generation was explained as coupling phase. That means between the two genes capital B and capital L, that is between these two genes, there is a coupling, gametic coupling. Okay, so this phase is known as coupling phase by B.Sc and Punnett. Okay, next phase is repulsion phase. So when they again cross blue flowers with round pollen grains with red flowers and long pollen grains, as usual in F1 generation, they got hybrid blue flowers with long pollen grains. This is a normal F1 generation. After that, when they test cross this F1 hybrid with the recessive parent, okay, so in F2 generation, the expected ratio is 1:1:1:1, whereas they got different ratio such as 1:7:7:1. So this is the deviation from Mendel's law. Okay, instead of 1:1:1:1 ratio, they got the different ratio. If we examine, so the blue long are with normal 1 ratio, red round also normal with 1 ratio, but the blue round and the red long, which are present in parental combination. So these two alleles, that is capital B and small l, capital L, they repel each other. Okay. So here the dominant alleles they repel each other and the recessive alleles they repel each other. So these 1:7:7:1 test cross ratio indicates that there was a tendency in the dominant alleles to repel each other. So these type of tendency of both dominant alleles or both recessive alleles to repel each other is called as repulsion phase. Okay. So this is all about the coupling and repulsion phase or the hypothesis. Next we will move on towards the types of linkage. So Bateson and Punnett could not explain exact reason of coupling and repulsion. They just did the crosses and get the F2 ratio such as 1:7:7:1 and 7:1:1:7. So they just did the experiments and got the deviations from the Mendelian ratio. After that, T.H. Morgan, another scientist, he performed experiments on Drosophila, okay, and found that there are two types of linkage based on crossing over. As we know that crossing over is exchange of chromosomal segments between non-sister chromatids. So this linkage is related with the crossing over, whereas it is inversely proportional to the crossing over. The more the distance between two genes, there is a higher percentage of crossing over. Okay. So based on crossing over, linkage is classified into two types. First is complete linkage and second is incomplete linkage. Okay. So this complete linkage is found in male Drosophila and females of silkworm. Okay. And complete linkage, it is a type of linkage where there is a complete absence of recombination. That means there is no crossing over. Okay. So this type of linkage is called as complete linkage. Second is the incomplete linkage or partial linkage. Okay. Incomplete linkage involves some percentages of crossing over. Okay. So there is a presence of crossing over and that linkage is called as incomplete linkage. It is present in female Drosophila, again maize and pea plant. So let's study the complete linkage. This complete linkage is present in male Drosophila. So in most of the organisms, crossing over takes place both in males and females. But in case of male Drosophila, crossing over absent. Okay. So in 1919, T.H. Morgan, they crossed grey-bodied and vestigial winged Drosophila with black-bodied and long-winged Drosophila. Okay. So in F1 generation, they got hybrid grey-bodied and long-winged Drosophila. Let's study the cross. So when a grey vestigial Drosophila crossed with the black longed Drosophila, so in F1 generation they got hybrid grey long Drosophila. Okay, so this is a normal one. But when this male F1 hybrid grey long Drosophila when back crossed or test crossed with the female black vestigial Drosophila, so in F2 generation, instead of 1:1:1:1 ratio, they got 1:1 ratio, that is 50% characters are grey vestigial and 50% are with black long Drosophila. That means in male Drosophila, in F2 generation of test cross, all the combinations are the parental combinations, such as in the parents grey vestigial is present. So in F2 generation also grey vestigial is present. Similarly, another parent having black long Drosophila. So similar kind of progeny obtained in F2 test cross. So in this way, at the end of test cross, only the parental combinations are present. So in male Drosophila, 100% parental combinations are obtained from test cross progeny. So the test cross ratio is 1:1. So why this is happen? Because in this type of linkage, these genes are closely associated with each other and they tend to remain together in next generation also. So only parental combinations are there, no new combinations. So there is no crossing over. Such type of linkage is called as complete linkage due to the complete absence of crossing over. Okay. Next is the incomplete linkage. See, the linked genes, they do not always stay together because of phenomenon of crossing over. Okay. So the crossing over is the exchange of chromosomal parts between homologous chromosomes. Okay. So these linked genes do not stay together due to the phenomenon of crossing over. So when the linked genes, they are present widely located in chromosomes and they have the chances of separation due to the crossing over, that type of linkage is called as incomplete linkage and the genes are called as incompletely linked genes. Okay, it means incomplete linkage involves crossing over, so new combinations are formed due to the crossing over. So these type of linkage present in female Drosophila. Let's take a cross. So in female Drosophila, when grey-bodied and vestigial winged Drosophila crossed with black-bodied and long wings, so in F1 generation they got grey-bodied and long wings as usual. So when F1 female hybrid Drosophila are test crossed with the male Drosophila having black and vestigial wings, then in F2 generation of test cross they got 4 types of offsprings. Okay, out of 4 types, so the 2 offsprings they show 83% of parental combinations and 70% show recombinations. Okay, so in this way, in incomplete linkage, there is no only parental combinations because new combinations are also formed due to the crossing over. So this type of linkage in which test cross results show that there is a 70% cases of crossing over occurred between linked genes. So this type of linkage which involves the crossing over is called as incomplete linkage. Okay. Next is the linkage group. See, linkage group, these are the linkage group gene 2, gene 3, gene 4. These all form the linkage group. So linkage groups are nothing but a set of genes at different loci on the same gene and they except for crossing over tend to be inherited together. So all linked genes of a homologous pair of chromosomes form a group and this group is called as linkage group. So in Drosophila there are 4 linkage groups. For example, in Drosophila there are total 8 chromosomes. So diploid in nature, so there are 8. So there are 4 pairs of chromosomes. So 4 linkage groups. Whereas man has 23 pairs of chromosomes, so they contain 23 linkage groups. Okay, so this is all about the linkage groups. So here is the significance of linkage. So one of the most important significance of linkage is it reduces chances of recombination. So it helps to maintain the parental combinations. Okay, it also plays important role in determining the nature of scope of hybridization and the selection programs which is beneficial in plant breeding. Okay, and the linkage between genes controlling 2 different desirable characters will help in simultaneous improvement of both characters. Okay, so this is the significance of linkage. So these are certain references for this topic. So friends, this is all about the linkage phenomenon. We will stop here and we'll meet in the next session. Thank you very much.